Today we will be talking about a virus known as Nipah. This is a virus many people have not heard of, mostly due to its prevalence in South and Southeast Asia and its rather recent emergence there. In case you haven't heard of it, Nipah falls into the Paramyxoviridae family, which includes measles and mumps, to name a few other viruses. It is a single-stranded, non-segmented RNA virus, and it is also enveloped. Although it was discovered in 1998, it is suspected to have originated in the 1940s. Some facts about Nipah. It can come from two sources, and generally it comes from bats and pigs. Specifically, in the case of bats, it comes from date sap. When studying Nipah in bats, the method of transmission is very different from Nipah in pigs. The consumption of date palm sap by these fruit bats is what can lead to the infection of humans. Infected fruit bats lick up the sap on the tree, and their saliva drips into the containers used to collect date sap, which contaminates the sap. Humans can become infected when they drink this sap. Today, we're flying over to Bangladesh to study the Nipah virus. The first known outbreak was in Malaysia in 1998, and outbreaks in Bangladesh and India have followed soon after. Direct bat-to-human transmission generally occurs through date sap, although other possibilities can include living under a bat roost. So when people collect date sap from the trees, infected fruit bats come for a snack, which unfortunately contaminates the sap reservoir with Nipah virus. Large containers are used by humans to collect the date palm sap, and although this is effective for humans, this also provides an excellent opportunity for the fruit bats to snack. Fruit bats often land on the tree and lick up the sap, which then eventually also trickles into the container. Bat urine and feces, which has also been found in the container, can also lead to contamination. Humans become infected when they consume the contaminated raw date sap, and this is the most common form of transmission, at least when it comes to bats. The Pteropus fruit bats are considered to be the primary reservoir for Nipah, and specifically the large flying fox and the small flying fox. Now, we're going to take a quick flight to Malaysia. Although the same virus, transmission here occurs through pigs. Pigs here are kept in large slaughterhouses, which is optimal for animal-to-animal spread of viruses. This is not the case in Bangladesh, where animals are kept in small groups. Infected fruit bats here shed the virus, which infect pigs here on the farms. This has been attributed to the fruit bats eating fruit, which then fall down to the pigs, who then eat the infected fruit. The pigs here act as an amplifier host, which means they accumulate high viral loads, which allows the virus to jump to humans, which does make this a zoonotic disease. The outbreak here occurred in pig farms in peninsular Malaysia, and specifically in pig farmers. People who got Nipah here were in close contact with pigs. Symptoms were initially attributed to Japanese encephalitis, although obviously this was later proven to be false. Epidemiological evidence suggests that the primary means of spread between farms and between regions was the movement of the pigs in between the farms, and the primary mode of transmission on pigs' farms was believed to be via the respiratory route. Nipah virus infection is associated with respiratory distress and encephalitis, with no efficient vaccines or therapeutics on the market. Patients who have a Nipah virus infection show symptoms 3 to 14 days after exposure, and these symptoms can start with a headache and drowsiness, and then it can quickly develop into respiratory symptoms, such as atypical pneumonia and sore throat. After these symptoms present, the patient can succumb to a coma within 1 to 2 days, with encephalitis being a critical complication in many patients. In various outbreaks from 1998 to 2018, the mortality rate has varied from 40 to 70%, which does sound concerning, but don't worry, we're going to give you a quick rundown on how your immune system can fight this virus.
The Nipah virus enters the body through the oronasal route, and then its G protein binds to a receptor called ephrin B2 or ephrin B3, which is expressed on smooth muscle cells and endothelium in the brain, lungs, and prostate. Cellular helicases recognize viral genomic RNA and activate the type 1 interferon response in antiviral genes. The type 1 interferons, IFN alpha and IFN beta, induce an antiviral state. They do so by inhibiting protein translation, degrading RNA, and inhibiting viral transcription and assembly. Interferon alpha and beta also increase the expression of receptors for natural killer cells and activate them to kill virally infected cells. Additionally, some of the virus is broken down and presented on a protein called major histocompatibility complex 1 or MHC1. These are neutrophils, a type of white blood cell whose main purpose is to phagocytose and destroy pathogens, as well as signal to induce inflammation and recruit other immune cells. One particular method of defense is the neutrophil extracellular traps, or nets. Nets are the release of DNA from the neutrophil's nuclei, which traps pathogens. While this kills the cell, it also releases several antiviral particles such as myeloperoxidase and alpha defensins. But the Nipah virus is sneaky, and it can actually protect itself from the nets. Remember that type 1 interferon response? Well, Nipah virus produces structural and non-structural proteins, such as proteins P, V, and W, that affect the activation of interferon type 1 production and signaling. For example, these nifty viral proteins interact with STAT1 and STAT2, transcription factors that help to induce the antiviral state that we just mentioned. So, no type 1 interferons makes infection a lot easier for our virus here. But don't panic, your body has a slew of other cytokines that prepares itself to battle the infection. To name a few, interleukin-6 induces dendritic cell maturation, IL-8 induces chemotaxis of granulocytes to the site of infection, and MCP1 regulates the blood-brain barrier permeability. All of these cytokines together promote functional monocytes and T-cell movement to the site of infection. As we continue to talk about the immune response to Nipah virus, there's a small disclaimer. Since this is a relatively new virus with a high mortality rate, there isn't a lot of clinical data from humans regarding all aspects of the immunobiology. There is one study that looked at 2 patients in Kerala, India that we can talk about. These patients had elevated effector CD8 levels and lower CD4 levels. However, total B-cell lymphocytes, activated B-cells, and plasmablast cells were all increased. Another factor of pathogenesis for Nipah virus is its ability to infect monocytes and lymphocytes. A study in pigs showed that monocytes, NK cells, CD6 positive, and CD8 positive cells all supported Nipah replication. Here's some good news for your T cells, though. They may already be able to recognize this virus. There are some viral epitopes that are conserved in the Paramyxoviridae family that result in cellular cross-reactivity. If a patient has already been exposed to a different virus in this family, like the measles or mumps, then their T cells have the potential to recognize Nipah-infected cells and offer more protection. Currently, there has been little research to investigate whether the adaptive immune response is simply inefficient at dealing with Nipah virus infection or whether the immune response itself is immunopathogenic. This would mean that the body is creating a large, ineffective immune response that exacerbates the pathogenic process.